Good evening, everybody. My name's Bob Hill. I'm the Executive Dean of the Faculty of Sciences and the Director of the Environment Institute. We've had a late problem with our MC, so I'm going to take his place and run us through tonight. So, and there also seems to be a small amount of confusion about whether the start time was 5 o'clock or 5:30. So for those that are here expecting it at 5, my apologies. We will start now, and if anyone comes in at 5:30, they can pick it up as they go along. So what we're here to do tonight is to launch the Australian Bioactive Compounds Centre, which is joint with the University of South Australia, and also the Centre for Conservation Science and Technology. And these are two very important initiatives for both the Environment Institute and the University of Adelaide. And I'm really pleased to see such a crowd here tonight, and thank you all very much for coming along. So first speaker we have this evening is Professor Andy Lowe, who is the Director of what is called COST, the Centre for Conservation Science and Technology. So welcome, Andy. Come on up.
Welcome. And what I would like to do is go through exactly what the Centre for Conservation Science and Technology is. So it's a new centre that's being established at the University of Adelaide. And we understand that there are a large number of problems with biodiversity decline, with impacts on ecosystems and ecosystem services. We've had a large amount of research and observation monitoring to understand that decline. But really, it's time to focus on the solutions to those pressing global conservation issues. And so the members of COST are a group of researchers that really are working at the applied end of conservation restoration, but applying science and technology to those types of problems. And largely, the focus of the group is looking at improving biodiversity and biosecurity monitoring and outcomes, improving conservation and restoration practice, and reducing wildlife and forest crime. The centre brings together 5 key capabilities, and some of these are parts of local initiatives, some of these are parts of national initiatives. The first key capability is the Terrestrial Ecosystem Research Network, which combines a range of plot-based monitoring with remote sensing, biogeochemical flux monitoring to really understand the state and condition of Australia's unique biodiversity and ecosystems. The second capability is a capability that's been built around drone monitoring and also extends into some local remote sensing. So again, this really is at the cutting edge of latest technologies that can be used for monitoring ecosystems. The third facility is around advanced DNA identification and forensics. So being able to extract DNA from difficult compounds, wood, for example, traced DNA elements, allows us to track the origin of biological products, and this is particularly relevant to wildlife and forest crime. The group also brings together a large number of study sites, whether these are biosecurity type sites or restoration sites, particularly around South Australia. And the group has exceptional expertise in a range of analysis, whether that be the middle one, trade routes analysis, analysis of the distribution of genetic variation, or really understanding the cost implications of different conservation or restoration approaches. So a combination of a range of different disciplines. The group that are brought together is a key group of 15 academic members, and there are 6 affiliates with the centre. Now, between this group have actually published over 600 scientific papers in the last 5 years, and brought in more than $60 million of funding for research. Now, $15 million of that is actually work with government and also industry. So the group are very applied and very interested in solving problems together with partners. Also, this group over the last 5 years have had between them 130 PhD students and 113 honours students. So it's a real powerhouse of conservation science and technology research. Also, partnerships is very important. We have a number of joint appointments with local partners around the state. So Department for Environment, Primary Industries, Research and Development, South Australian Water, and also other local NGO organisations. We also extend partnerships to a number of key international organisations. And just signed this week is an MOU with Conservation International, and David Emmett will be our guest speaker who will talk about that in a minute. And we have a number of other significant interactions and partnership agreements with a range of other international partners and a range of international national partners as well. And between the group are working across 55 countries. So the work, you know, the mantra, which is working locally, extending globally, really applies to this group. And I think what we need to keep in mind with the group is what's able to be done by bringing together this type of group. And for me, the context of the Intergovernmental Panel for Biodiversity and Ecosystem Services and really understanding what the value of nature is a critical component that we have to get engaged with if we're to solve these types of global issues. And one project that the group has been working on and brought together is to look at how restoration can actually provide additional benefits. So restoration of native vegetation, how that can provide pollination services for crops, but also some of the health benefits that I don't know if you saw Phil Weinstein's talk earlier today, how that can also have associated human health benefits. So by understanding the dynamics of systems, by understanding how the integrity of ecosystems relates to these types of services, and then being able to value those services, then we believe that COST can really help solve some of these global biodiversity issues. So with that, I'd like to launch COST. And I'd like to invite up David Emmett, who is Pacific Director of Conservation International. And David has agreed to help launch the COST Centre and to help provide a global context to some of the key conservation initiatives and issues that face us today. Thank you, David.
Thank you very much. It was great to hear the work that you're doing and are planning to do and how closely it's aligned with the work we're doing. It's very reassuring since we have just signed an MOU. So I wanted to talk a little bit about the role of science and technology in conservation. Now that could go on for several months, so I was trying to hone it in a little bit and focus on the sort of activities I think we could do together. First of all, some of you may not have heard of Conservation International. Some people have referred to us as the NGO that nobody has heard of that's done the sort of the best work. Usually it's our team who say that, but we do have a very strong board with people like Harrison Ford and the CEO of Walmart and so on. So we have some very big global partnerships. And globally we have nearly 1,000 people. I'll move to the area that's interesting for us. In the Asia-Pacific region, we have about 300 people. We're working in a series of key major geographies. One of them is the Greater Mekong, one of them is Indonesia, and one of them is the Pacific, working under the framework of the Pacific Oceanscape in a partnership with the Pacific Island Forum. And we have, as you'll see from this previous map, maybe, we have a very significant presence in this region. There are countries where we have an office and a country where we work through partners. And our work is very much partnership driven. So we have an annual budget in the Asia Pacific region of about 30 million, of which about 16 to 17 million we give away as grants to partners, whether it's the government, local NGOs, local communities, academic partners, or wherever. I'm based in Singapore. I used to live in East Africa and then spent nearly 9 years in Cambodia, and then I was given this responsibility of overseeing the Asia Pacific. But with that responsibility came the move to Singapore, which was, I thought at first, was a bit of a less interesting country as a sort of a biologist by training, but it's actually quite fascinating. And it does also have great corporate connections and academic connections. That's where I met Lian Pin through the University of Singapore, which has now brought me here to this partnership, which I think is going to be very fruitful. So Conservation International, we are nature-focused, but we are focused on how people need nature. It's about the interaction between people and nature. It's showing that the ecosystems provide services that we rely upon. And the basic premise is that nature doesn't need people, people need nature. And if we lose these ecosystem services, we basically are destroying the planet for our children and our children's children. So we're looking at sustainability processes. Now, I went back over the course of my lifetime. I'm not particularly old. My hair's going gray, but I'm only in my early 40s. And I went back through the technology that I've dealt with in conservation in the past. Looking at modern day with satellite imagery, we've just recently got access to the full NASA files of all satellite imagery they've got. They needed to show that they were giving something back to conservation, so we've been given access to that, which is incredibly high-resolution data. The European Space Agency has just given, I think, free data for globally. So we now have the ability to basically use that information anywhere in the world to map ecosystems, to model, look at fire predictions or whatever. Going back about 15 years, this was me in the mountains of Cambodia with the most up-to-date technology at the time, which was a GPS, teaching the rangers how to basically use a GPS to find their way around in the field. Going down here to a site in southwest Madagascar, probably almost 20 years ago, we were asked by WWF to do a survey in the area and map out the different habitats. Nowadays, you can use satellite imagery, radar imagery, etc., to identify different forest types. There we had to hike over every single mountain to see what was on the other side and map it all out by hand. And then the last image, I can't show you because we didn't have digital cameras at that time because the technology didn't exist. So what I want to do is quickly focus on 3 areas that I think would have extremely interesting opportunities for us to work together. And these are areas where technology has really helped advance conservation science globally and especially in the Asia Pacific. And I'm going to give a few examples from the Asia Pacific. So remote sensing, you were mentioning about satellite imagery and drones. We have a partnership with the facility here on drones. So there's a team who goes to work with our organization in New Caledonia, and it's using drones technology to actually identify the dieback of kauri in the forest there, because you can see the disappearing and the changes in the tree canopy. Some of these places you can't get to because they're inaccessible or they're cultural forests where we're not simply allowed to go. Monitoring. This on the left, have some of you heard of the Phoenix Islands Protected Area? PIPA, it's part of Kiribati. So Kiribati made a very large area of their territory off limits to tuna fishing as a plan for sort of restoring the sustainability of their tuna fishery. This area on the left was in October 2014. It was put in place, and as you can see, rather than us actually having to go out there and do patrolling, we use these the vehicle monitoring devices, the vessel monitoring devices, and you can see that the protected area has been properly respected. So this is just a couple of examples where satellite imagery has really massively moved forward our agenda in conservation. And it could be RED, it could be helping a country to transparently show its forest cover, because what we see as forest cover in Indonesia and what the government sees as forest cover in Indonesia can be quite significantly different percentages, because they count certain things as forest, such as an area that used to be forest, whereas we see that as degraded bare land. So using satellite imagery, it's a transparent process. Looking at forest cover change, in the southwest of Cambodia, down on the bottom left here, I would try and use this as a laser, but I think I'd mess it up. But you can see right in the middle of the map, the central cardamom's protected forest. We set this up as a protected area. And over the course of more than a decade, a lot of donors have been putting money into that, and they want to see what impact we've had. And we can actually show from satellite imagery and from showing forest cover change that the surrounding areas have seen significant degradation, whereas the actual central cardamoms where we've been working has seen about an order or less of magnitude different forest cover change. It's been a very, very successful program. Other technologies that we have in protected area management, camera trapping, DNA analysis, drones, etc. There's a huge amount of technologies we use now. I remember going into the mountains in Cambodia and taking the camera traps and winding the film back and finding that you had 36 photos of a tree branch moving in front of it, and that's it. And all of that effort hiking into the mountains was gone. Nowadays, you've got digital cameras that can take thousands of photos, and I think soon, if not already, the technology will be there for them to be uploaded via drones and then downloaded to your computer. It's a dramatic increase in the efficiency of conservation, and the important thing on that is that every dollar counts in conservation, and the numbers of dollars going into conservation is actually dwindling, not just from governments. I think Australia is actually going to be sort of increasing now. You have a new government in place, but there's just a dramatic decrease in investments in conservation around the world. So we need to show bang for the buck, and that really comes from cutting-edge technology. What we've set up here is a partnership with Hewlett-Packard, because with thousands of camera traps in the forest comes an awful lot of data, and that can sit on hard drives and no one has time to go through it. So we've developed a software tool that allows us to go through camera trap photos and sort it, not only show what the species presence, absence data are, but showing trends in wildlife in different protected areas in networks around the world, and then giving us a guidance as to why these trends may be happening. And this is really unique, and the scale of data management is something we just simply couldn't do in the past. We do some very exciting species tagging and tracking work. I'm just going to skip to this slide. This is in West Papua. There's an area where over the years we found that the local fishermen were catching very small fish, and the whale sharks had learned to go up and feed from the nets. We thought this was fascinating, and we put it on YouTube and got lots of hits, but that didn't really do an awful lot for conservation purposes, other than get people very interested in that site and then increase the tourism potential. What we did find, though, is occasionally these animals would swim into the nets and get caught. With the advent of satellite radio telemetry, we've been able to do something very, very exciting on this. I'll wait for him to finish feeding. He could be there all day, actually. So what we did was when they were, this one here you can see is scooped up in the net. You can't quite see the scale, that's more than 7 meters long, but we got in there with it, or my team did, the lucky field biologists. And as you can see on the top right, we attached some very, very innovative satellite tracking information that sends real-time data with just less than a second of it breaking the surface. And down here is the area in West Papua where these whale sharks have been tracked. This is only in the course of, this is like 4 months ago, something like that. We found that they've been going down to Papua New Guinea. We found that they've been going to feeding sites we didn't know about. We found that they've been breeding in certain sites that we didn't know about, and the government has then changed the entire protected area system in that area to ensure they're protected throughout their range. Something that you could only dream of even 15 years ago. I'll get past him again. Same thing with manta rays. We were tagging manta rays in Indonesia because we were finding that there was anecdotal evidence that they were getting hunted. So we tagged them in, as you can see down here, in Bali. Anyone who's been to Bali, there's an enormous amount of tourism around manta rays. We tagged them, find that they were moving over to Lombok where they were being hunted, and smaller numbers are coming back. So the government was putting millions of dollars into tourism around manta, but they weren't protecting them throughout their range. That simple study and the analysis of the data led to the government in Indonesia making a blanket ban on manta ray hunting across the whole country. So it's a really interesting example of how technology can guide our better understanding on species and can help protected area management. We also, by the way, had to show before they actually made that commitment, we had to show the economic benefits of conserving manta rays, that they would get about $1 million per manta during the lifetime of the manta from tourism, because if you don't put a dollar value on it, sometimes there's not quite so much interest. Other areas that we use technology is in conservation planning. Just recently we've been working in North Sumatra and now in West Papua. The government in West Papua has said they want to set it up as a conservation province. It's 92% forested, and they said, can you work with us to show us which areas should be protected? We want to ensure that at least 80% of the province is remaining as forest, but we also want to see economic development. So we can use the latest technologies and science to model the areas that are best for certain crops. We can look at the impact of climate change and see where coffee will be grown in the future. We can demonstrate all of the interconnectivities between the forested areas and the reefs. We can look at the flow of toxins into the sea, and we can look at the effects on people. We can look at the effects on fisheries, and we can do a huge amount of modeling. And with that, we can do what they call a sustainable investment action plan. And the government of Indonesia has tasked us with doing that for West Papua, to look at corporate investments, palm oil expansion, etc., and see that it can be done in a way that respects nature and allows economic growth. Now, that's an opportunity where I think we could have a great deal of partnerships with the university here, because we're talking about an entire province that has been presented as an area where there needs to be strong activities in the next couple of years before the government re-elects somebody else there who may have different plans. Another thing that we sometimes forget about is that technology, it can be a double-edged sword. You can put some information online, and people can put information online that is not necessarily true, and then the information gets out there very quickly. But it's a very, very good way of sharing conservation success. And also, if you develop a really great tool that allows you to track coffee prices in the market, that's not very useful unless the local smallholders in the middle of North Sumatra or wherever have access to it. And nowadays, they have it through their mobile phones. There's more internet coverage globally now than there's ever been, and there's pretty much nowhere I've been to in the region where you don't get that. So this is a really big tool that we need to be very careful of how we use it. Another example, I think, is the work that Greenpeace did a couple of years ago, highlighting the activities of Unilever, I think it was, or Nestle, and the non-sustainable palm oil that they were selling. Now, that led to a behavior change in the public, which led to a behavior change in the company, which eventually went all the way back. And then the president of Indonesia has now stated that there will only be sustainable palm oil grown. There will be no more deforestation on peatlands, and that there will be no more, well, allegedly no more deforestation and burning. Although when I left Singapore, I could still smell the wood smoke from Sumatra. So these things take time, but it is happening. And I think that the internet is a technology that we really have to seize to our advantage. Of course, it's a double-edged sword, because nowadays when we send rangers into the forests, the guy down living next to the ranger station will call his friends in the forest and say, watch out guys, they're coming this way. So, you know, with anything you develop a new technology that allows us to improve patrolling, it can be used from the other side as well. One thing I just wanted to show as well on the side of technology and internet and raising awareness, personally I think this is a very unique time for conservation globally. After the meetings in Paris, we've seen an unprecedented investment by governments, and I think the public are finally realizing that no matter how wealthy you are, how disconnected you are from what's happening in Brazil or Indonesia, you still live on the same planet. And we all have realized that. And we're seeing it increasingly from individuals and companies and governments, that they're all buying in to the concept of better environmental best practices. We produced a series of short videos that have been very, very popular, and has really helped people to start a conversation about the environment. I'll just play one for you now.
Some call me nature. Others call me Mother Nature. I've been here for over 4.5 billion years, 22,500 times longer than you. I don't really need people, but people need me. Yes, your future depends on me. When I thrive, you thrive. When I falter, you falter. Or worse. But I've been here for eons. I have fed species greater than you, and I have starved species greater than you. How you choose to live each day, whether you regard or disregard me, doesn't really matter to me. One way or the other. Your actions will determine your fate, not mine. I am nature. I will go on. I am prepared to evolve. Are you?
So to conclude, the Asia-Pacific region is extraordinarily important to the rest of the world. The deforestation in Indonesia is going to have an impact on climate change. What happens in the Pacific is going to have an impact on tuna stocks the world over. So what we do is extremely important, and now is the time. So this is why this partnership is so exciting, because we're all working towards a common aim, and we've all got really specific interests and skill sets, and I think the sum is greater than the parts. So working on partnerships like this is extremely exciting. So I'm very happy to forge this and to see where it leads. Thanks.
Okay, thanks very much, David. And I'd personally really like to thank David for making the time to travel down for this launch. It's a great honour for us and a real thrill to be able to partner with a group like Conservation International, and I think it takes us to a whole new level in what we can try to achieve. So thanks very much. Now we move on to the launch of the Australian Bioactive Compounds Centre, and in order to perform that launch, I'd like to introduce Professor Phil Weinstein from the University of Adelaide and Associate Professor Bob Milne from the University of South Australia. Thank you.
Thanks very much. Very simply stated, the aims of this centre is to use ecological and indigenous knowledge to discover bioactive compounds in arid zone plants. Now, why arid zone? Because arid zone plants are stressed more than any others by temperature, drought, sun, salt, everything. And that increases the probability of them containing some useful compounds that we might be able to use for a whole variety of purposes, from cosmetics to new antibiotics to anti-germination or anti-feedant devices in agriculture, antifungals, whatever. People have looked for these things obviously in the rainforests, they've looked for them in the ocean, but the arid zone, partly for geographical reasons, I guess, remains largely underexplored. So it's the aim of the centre to use our ecological knowledge and our indigenous knowledge to try and capitalise on the environment that we're in and the opportunity to develop some of those into commercially useful products. What have we done so far? We have at both universities, I'll hand over to my co-director Bob in a minute, but at both universities we've gained centre approval and we've done a positioning literature review. Brad Simpson did this for us, and those of you who were at the conference to hear his presentation earlier today will have heard a very eloquent presentation of the justification for targeting the arid zone and for using an ecological rather than a mass screening approach, as has been done by some drug companies. We've also had a first sort of integrated expedition, which we've followed through from the ecological and systematic knowledge of plants in the area to collect some things that might have contained some likely candidate compounds, extracted those and screened them as a proof of concept more than anything else, without the greater hope of finding anything. It was really just to demonstrate to potential industry partners and colleagues that we could collaborate and go through that whole plant to product stream fairly efficiently as a team. And already we've got a potential lead compound out of that demonstration project, I guess, which is very exciting. We have a couple of industry partners that we're already in discussion with. Some members of the centre have pre-existing relationships that we capitalise on, and thank those members obviously for contributing their collaborations to the centre. A few weeks ago we appointed a postdoctoral research fellow, Casey Hall, who trained at the Griffith, and those of you who know Griffith had a big project with AstraZeneca for looking for bioactive plant compounds. So welcome to Casey. She's very much chief cook and project manager for us. So without her, little would progress. And that really leaves just the official launch, which is what we're here to do today. I did just want to highlight, though, the quality of the collaborators. So we're doing this jointly between Adelaide University and University of South Australia. And the reason we're doing that is because we've got well above world standard expertise in all of the requisite areas between us. So we've put our sort of, Vice Chancellor's differences aside and thought this is a really good thing to do for South Australia. And we've got, just as two examples, we've got ecology, University of Adelaide and ERA 5. We've got others, but obviously very relevant to this centre. And again, pharmacology and pharmaceutical science, UniSA, they have others, but very relevant to this centre. We're putting those skills together. But rather than talking about those skills, I'd like to present one of our collaborators, Professor Birger Møller from the University of Copenhagen. Before handing over to him, though, as in terms of quality, Birger won't mind me advertising the fact that he's one of the world's top 10 plant biochemists, and we're delighted to have him fly down as well for this event. And thank you very much in advance, Birger. But before I hand over to you, I'd like to ask Bob to say a couple of words as well. Thanks, Bob.
Thanks, Phil. Yes, I'm from the School of Pharmacy and Medical Sciences, University of South Australia. And about a year ago, Phil approached the head of school with the idea of setting up a centre. And he approached our school, I reckon, because he's had long collaborations with one of our researchers in the school who's been doing a lot of work looking for compounds, bioactive compounds from the Australian bush. And this person, Dr. Susan Semple, has spent many years establishing a very trusting and sensitive relationship with Aboriginal community in Cape York, and using their knowledge and skills to identify different plants which they've used for therapeutic purposes over centuries. And with that knowledge, she's then been able to take these plants, different portions of plants, and look for extracts or take extracts and test them for different pharmacological activities such as antiviral, antibacterial activity, anti-inflammatory activity or anti-cancer activity. And then those which possess promising activity have been, active components have been isolated from them and then identified using mass spectrometry, HPLC, to look for purer compounds which of course are likely to be of greater consistency in terms of their activity. So that's where the University of South Australia comes into this partnership. I think it's going to be a fruitful partnership. Phil and I have known each other for quite a few years, and I look forward to its continuing success. I'm pleased you initiated it, Phil. Thank you.
Thanks very much, Paul.
Thank you for the invitation to come here and say some words. And I really enjoyed being here 2 days. I think Australia, many times you think you are far away from everything else. But coming here and then seeing how much work is being done within this area, it's really impressive. I don't think it's because you have had that much funding, but you have people who are engaged and who want to solve problems. And that's really the driving force which counts. So when I come here and then I learn that you have this center, that's super, because doing this type of work, it's highly, highly interdisciplinary if you want to be successful. And to have as many people as possible involved and people who actually want to be involved, that's the key to success. So really to see this sort of joint work between the University of Adelaide and University of South Australia is really, really great. I think that's one key parameter to success. I have always worked with plant insect, plant microbe communication, and how do plants communicate and how do they try without sort of being able to move. And now as a head of Centre for Synthetic Biology, we are trying to use that knowledge, these building blocks, these modules you gain, to put them together in new ways to design molecules, products which are to the benefit of society. In the other center, we work on how to make plant robust. What make a plant collapse? When it can't cope with things anymore? And when you do that basic science, you get knowledge which you can use actually to do proper synthetic biology, where you use third generation molecular biology, but you do it right so that you get the consumer support and you make products which are valuable. Politicians talk all the time going towards a bio-based society, but they don't fund that very, very heavily. I mean, it's something, it's a nice statement, but what's inside? And I think one technology which can help to do this is really third generation genetic engineering. I'm actually not a fan of genetic engineering. If you just do things and try and well, this didn't work, then I try something else. It's really knowing what you're doing in advance based on solid information, knowing how modules in nature work. So that's a key success parameter in our synthetic biology center. Then there can be a lot of disruptive technologies here, and I've tried to give some examples. When you try to understand how plants work, then you also get new industries built on that in a very profitable and beneficial way. And it's when you work with synthetic biology, you really don't harvest nature. You need very little plant material to do your analyses, and if you think well about it and substitute industries you are sort of laying down with new industries, then ecology works out fine. You get new patenting ideas by doing this type of work. If you want to avoid a patent which really irritates you, do some ecology and find out who else does that reaction in a different way which is not covered by that patent. And you get many examples of that. And then get things done right so it's accepted by the community. We only use genetic engineering when it's contained, and we use the koala eucalypt system to visualize climate change, because the koala, you know what it eats, you know what comes in, what goes out, you know the eucalypts, you can measure how they change their constituents with climate change. And so that's really using that iconic animal. It's to get focus on climate change in the general public. Of course, the koala is not sort of an important animal for production or anything, but it's an iconic animal. It's something you can get people's attention to. And then you have a lot of opportunity in Australia. Of course, you are facing huge challenges with the climate change. It's tough, but you're also ahead of many other places in addressing these issues, and that really makes you a very good collaborators. So I think when you start such a center, and I'm saying that from this plant plasticity center where we work very interdisciplinary, and the nanoscience center where we work from the single molecule to plants on the field. When you do such a broad approach, you need funding for a long time. You get young people involved, and they engage themselves, but then when it's difficult projects, where it's difficult to get publications out, interdisciplinary research, you have journals like Science and Nature, they always have one referee who does not understand one of the topics, and then you are out. So there are many sort of disadvantages to work in this area. So a strategy really is about funding, and have funding for 10 years when you start such a program, to make sure you also harvest. The Australian advantage is really the fauna and botany you have here. I mean, the plants you have here are amazing. And the robustness is really about getting the proper funding organized. Then you will be successful. So when you're doing ecology, I mean, and genetic engineering and new technologies, you're turning things upside down. And a painter can get a lot of easy successes by doing that. But turning upside down science is very, very complicated. So that is sort of a warning that it is more complicated than you think. I'm only saying this because I'm much older than the average of the people doing this here. But I'm being surprised all the time how many difficulties. You don't have technologies, you need to develop them, you don't have model plants. You need to do a lot of things yourself. And then when you do something which is different, then you're not copying what everyone else does. And then it's more difficult to understand and explain and justify. So it's really it's exciting, but it's goddamn also difficult many times. Success parameters, what are they? That's collaboration and openness. I mean, I developed this principle. I don't want to be cheated 3 times, but I don't mind being cheated twice. So then sometimes you get a lot more back than you actually give. And then focus on people who actually want and who are able to collaborate. Lots of people want to collaborate but doesn't know what it involves. And you should get rid of those as early as possible. And this system here is not based on a university merger. And I like that because university mergers is normally when things don't work and so on. Here you are doing something because you have people who want to do it. And that's a completely different, then you don't need to operate under the radar to avoid all the administrators getting involved. Then trust is very important, really important. If you trust people and you can talk to people, and also find the companies you can trust. There are absolutely many trustworthy, companies where you can tell them everything and you get lots of good communication. Then there are companies you should avoid. And so that's also make the right choices, and then mutual respect, and of course you need to be competent. Then how do you direct such a center? Well, I think you inspire, you prioritize, and you decide. But it's all the same thing, and that's when you get people in to help you, they don't understand that. So if you combine inspiration, prioritization, and decision making, then things run very smoothly. And when you're head of a center or you run a group, don't always argue if people don't agree. I mean, you should be ready to argue, and those people who question you, they are actually the people you should value. And then don't tell anyone that you're the boss, because then you're not the boss anymore. So you need to attract brains, and that's to create this vibrant environment where people want to come. And of course, PhD students should go out. We enjoy having several. We have 2 Danish PhD students here right now, and it's really good to have people out, and hopefully they also want to come back then. And then communication and to be able to listen. I mean, we do have 2 ears and only 1 mouth, so listening to what people tell you is actually that guides. If you get those comments early on, then you don't develop a product and then get irritated when people don't like it. If you get that communication up front, you really gain a lot. In the Plant Plasticity Centre, as I said, we work from single molecules all the way through to what's on the field, how are plants being drought stressed, how do they survive, and how do you develop production systems. Just one thing is an almond tree here. Almond trees flowers when they have had a certain number of cold hours. And there are compounds, secondary metabolites, which regulate that sort of bud break and flowering time. And they are cyanogenic glucosides. They are turned over at that stage, and they give a cyanide response, and that's a triggering for flowering. So now in the Mediterranean countries, you don't have enough cold hours, and what do? You spray with hydrogen cyanamide to substitute the cyanide, which is produced internally. And you can see how you sort of adapt in very, very strange ways, because then you're spraying with a toxin instead of having the plant doing the job itself. So there are many ways now when we understand this process, how do you control and how do you breed then for different levels of hydrogen cyanide, different turnover rates, and so on. So there are many aspects where you can gain knowledge. I mentioned this morning with the Møller joint work we are doing, and that's done with people in Western Australia and here and in Melbourne, with each bringing in their competences. Phil, we just mentioned Susan Semble, who has a strong sort of collaboration with the Aboriginal people. And then there are botanists and genetic engineers, or sort of molecular geneticists who are working on the Haemophilus. And we would like to do the content of these bioactive natural products and see which ones we can use and how you can do combinatorial biochemistry to make even more important compounds. When you do an initial screening of Haemophilus, I mentioned this morning that all these really complex molecules here, they're made from one single molecule, a C20 linear molecule, and two enzymes mix sort of these core structures, and then they're decorated by hydroxyl groups. These molecules will have very, very interesting properties. And plants who are working or living in such an environment, they really have to prioritize how they spend their energy. So they may not make very much of this, but they make sure that they make compounds who really hit their enemies. And those enemies are like microbes. It's the same enemies which human have when you want to do disease treatments. So I'm quite sure something very nice will come out of this. And then how do we produce these molecules? That's, again, I mentioned this morning that you can have photosynthetic systems where we have nanomachines, we know how they work, we can link the energy output of these nanomachines to the energy input to make these products, to make more of them. And the idea about in 10 years, I hope we have glass fiber houses like this when you walk in the park, and then you produce the valuable molecules you are looking forward to have in a very benign, environmental benign way. So I think this center is really spot on to get the collaboration going and exploit the opportunities. I look forward to look at the Haemophilus. Where are they producing these compounds? Which cell types are doing the jobs? That's the key to success, to elucidate these pathways. You can, if you find those cells, like here for fosculin, the coleus fosculi, it's 10 cell layers in the tiny roots, which makes these molecules. You can cut them out with a laser. You find out which genes are expressed there. You can do proteomics, and it's not that complicated to get the pathways running, so you can produce the compounds in yeast or photosynthetic cells. So I showed this one this morning here, and it's if you think about how diterpenoids are used now, they're used in herbal extracts, and it's only because they are so difficult to isolate. Plants make a whole battery of these. When you do it this way, then you only get one compound. It's a lot easier to isolate it. So the molecules are made here in the chloroplast, and the chemistry is done over here. And we call this guy Fred because he's thriving from fluorescence energy transfer, and he's really being energized. And then we have the nanomachines here, Nana, and the only thing you need to do is to bring these two together, so they live in Fred's house, and then they produce a lot of outcome. And this is what this is about. But you always need to know what is the input here and what is the output, so that you don't get side products. You know what you're doing. So you need to know all those modules. And synthetic biology, when you work with modules, it works. If you work with single genes and you just put a gene in and try to see what happens here, then no wonder you don't get the result you expect. You need to know how the modules work. And then we want to do the production, and we look forward to the collaboration here in Australia. And you can do a lot of combinatorial biochemistry here to make compounds you don't even think about exists. So there will be good opportunities. This is a syngenic glucoside, and I just mentioned that the control of flowering time in cherries and almonds are decided by these molecules here, by their ability to liberate hydrogen cyanide. We also made mutants in sorghum, which do not accumulate these toxins when they're drought stressed. If you plants, when they're really stressed and they don't have these molecules, then they will not be able to take up the water and reduce and the nitrate and reduce the nitrate to ammonia. So there's a lot of interplay between secondary metabolites and primary metabolism. Another example here, which was shortly named, also mentioned at the conference, this is another cyanogenic plant, that's a lotus plant, making these molecules here. You have an insect, the Zugina moth. It only feeds on plants making these toxins, and this is an insect. And just to see the sort of integration here, so when this female wants to mate, it sort of makes a plume of hydrogen cyanide around her to attract the male. So that's a male attractant. Whereas the male doesn't care, he just flies to her. If you look at the choice of her partner, then the female virgins have this average level, the male virgins this level. And then when you study these mating types, they start mating at 4 o'clock in the afternoon, they sit together until next day. So you can easily follow who are the mating pairs. And you will see that the rejected males are those which have a low content of hydrogen cyanide releasing molecules. And during the mating, the male transfers these molecules to the female, so she has a higher content of these toxins to protect her eggs. So all these levels are very, very intimate. If she cannot get enough of a hydrogen cyanide, either from the mating or from feeding on the plants, then she has a de novo pathway to make these molecules. And when you do that, when you find out the genes here, then the gene sequences are very, very different. So if you want to avoid a patent now how to use these compounds, then you just take the genes from the insect, doing exactly the same molecules. So there are opportunities here and there are difficulties. When the larva is attacked, it excretes bubbles here, droplets with these toxins, and they can withdraw them again to save them when the attack is gone. So it's a very complicated, and here you can see the pathway families, the enzymes are the same, but the gene sequences are completely different, so you can avoid patents. The eucalypts and the koala is really interesting again. I mean, how can you have an animal feeding on such a toxic diet? And many of the compounds here change. There's not a strong diurnal change in the volatiles and content in eucalyptus leaves. But if you have a heat wave, the content changes a lot. And so many of these compounds are hard to digest. So average passage time might be 7 days of eucalyptus material through the body of the tiny koala. These keto groups here, they cross-link the proteins. If you have a heat stress, then there will be more of these molecules here. That means that the feeding deterrence in the eucalyptus will be high. So there are a lot of things you can study and try to remediate here. The appendix of the koala is huge, and that's where the metabolism takes place. It's done by the microbiome, all the microorganisms who are there. And we are doing sequencing of all those together with Phil Hugenholtz in Brisbane to find out who's doing what. The male has a pouch, and that pouch has degradation products of the cyanogenic glucosides. And when the male climbs a eucalypt tree, it rubs the pouch against the trunk, and then other males knows that there's someone sitting up there. So these signaling and communication between the eucalypt and the koala are really, really interesting. This is a painting from Van Gogh, and as I said this morning, the sun is for Van Gogh the good God's son. And Van Gogh was stating all the time, humanity is harvesting instead of sowing. And I think now that sort of saying really comes through now, that we should sow instead of just harvesting. And I mentioned this again this morning. We need to explain what we're doing. Like Van Gogh was challenged with the quality of his paintings. Researchers are always sort of challenged with, are you doing things right? And be able to argue your case. And a genetically engineered mermaid is very, very popular. But if you call her a genetic engineered mermaid, then you sort of sculpture her in a different way. And so there's a lot of communication which we have to do better. So I wish you all the best in your center. I look forward to collaborate and good luck, all of you. And it's great this has been done. And I don't think you realize how far ahead you are from many other places in the world. So congratulations.
Look, we've had two fantastic talks tonight. It's a great way to launch these two centres. It's also a great advertisement for just how wonderful this NRM conference has been, that we're kind of just over halfway through. I think this is a breakthrough moment in South Australia, and one we'll look back on as a time when some very important things happened. So I think it's now that the balloons are released from the ceiling, or maybe not. We'll consider this event almost closed. I believe that we have food and drinks somewhere close by. Is that up in the foyer? So please, if you've got some time, stay and enjoy those. And we'll be pleased to talk to you if we get the chance. So once again, thanks for coming along tonight. I appreciate it. And the centres are now up and running. Thank you.